Hi, Assalamualaikum. My name is Siti Nur Hanis binti Ali, S52482 from Bachelor in Applied Sciences of Fisheries. Today, I will present my assignment of DNA marker in Fisheries Management for Biotechnology and Aquaculture course. However, first at all, we must know what is DNA. DNA is a short term for deoxyribonucleic acid (DNA), a complex chemical that carries genetic information for human and other organisms including fish. It was located at cell nucleus and mitochondria. Each strand of DNA in the double helix can serve as a pattern for duplicating the sequence of base. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar, and phosphate are called a nucleotide. It is a double helix formed by base pair that has information in DNA that stored as a code. DNA base pair up with each other. As you can see from the video, adenine will pair with thymine while cytosine will pair with guanine. This pair will form a unit. The order of all sequence of this base was like alphabet that can create the words and words that can create a sentence. It determines the information available for building and maintaining an organism. A marker is a DNA sequence with a non-physical location on a chromosome that was used to track the inheritance of a nearby gene that has not yet been identified, but whose approximate location is known. A gene or DNA sequence with a known location on a chromosome that can be used to identify individual or species. It also can be described as a variation that can be observed. There are variety of types of genetic marker, and the genetic marker serve as a signpost. This signpost will help researchers to relate a particular kind of threat or disease that they are trying to figure out the genetic base of to a particular location in DNA. So, to overcome problem associated with morphology marker, the DNA-based marker have been developed, and there are properties that must be fulfilled to be a good DNA marker. First, markers should exhibit high level of polymorphism. In other words, there should be variability in the marker. It should demonstrate measurable difference in expression between thread types O and gene of interest. 2. The marker should have simple inheritance. What does this mean? It means there should be absence of intra-local locus interaction. It can help in identification of heterozygotes from homozygotes. Next, there should be multi-allelic. It is useful in getting more variability or polymorphism for a character. Then, there should be absence of epitasis. It makes identification of all phenotype, homo or heterozygotes easy. 5. The marker should be natural. What does it mean for being natural? The being natural means the substitution of alleles at the marker locus should not alter the phenotype of an individual. Lastly, a DNA marker should have no effect of environment. Markers should exhibit minimum paleotrophic effect and insensitive to environment. Now, we have to know that there are two types of DNA marker. First, nuclear DNA and DNA, nuclear deoxyribonucleic acid. This DNA contains within a nucleus and of eukaryotic organism and highly conserved with a slow mutation rate. In mammalia and vertebrata, nuclear DNA encodes more of the genome than the mitochondrial DNA. and is composed of information inherited from two parents, one male and one female. This type of DNA marker is the most common DNA used in forensic examination. Next, we have mitochondrial DNA, mtDNA. mtDNA that are not highly conserved, but they have rapid mutation rate. It is useful for study the evolutionary relationship of organisms. Biologists can determine and then compare the mtDNA sequence among different species and use the comparison to build a evolutionary tree for the species examined. And because mtDNA is transmitted from mother to child, it can be a useful tool in genealogical research into a person's maternal line. So, why genetic markers are important, especially in fish? This is because mtDNA has a number of applications in fisheries biology, management, and aquaculture. In the past 15 years, mtDNA has attracted a lot of attention in many species, especially for population and evolutionary study. It has become a very popular marker and dominant genetic study designed to answer questions of phylogeny and population structure in fish for more than a decade. mtDNA studies particularly can contribute to identification of stock and analysis of mixed fishery, provide information on hybridization and introduction between fish, serve as a genetic marker in forensic analysis, and provide critical information for use in the conservation and rehabilitation program. The commonly used techniques are extraction fragment length polymorphism (RFLP), random amplified polymorphic DNA (RAPD), and plate fragment length polymorphism (AFLP), and microsatellite. First, application of DNA marker in species identification. The interspecific genetic divergence established through the species-specific diagnosis molecular marker provide precise knowledge on polygenetic relationship and also resolve taxonomic ambiguities. Since the structure of mitochondrial RNA gene, tRNA and rRNA, and the functional molecule of the 60S rRNA are highly conserved among the animal taxa, so the animal taxa they are related even distantly. So even a chance of a few nucleotides in such a gene between the related taxa might indicate a substantial degree of genetic divergence. For example, a specific diagnosis allogen loci were used for different species. For example, a specific diagnosis allogen loci were used for detect different species of trout. Species-specific diagnosis RAPD fingerprint are also were generated in several fish species and their taxonomic relationship has been analyzed. Next, the use of the markers in genetic variation and population structure study in natural population. Some of the possibilities are assignment of migrants, determination of genetic bottlenecks, effective breeding population estimate besides genetic variation and differentiation estimation. Impact of migration and gene flow on genetic differentiation also depend upon effective size of preserving population and number of migrants. Several evolutionary forces affect the amount and distribution of genetic variation among population and thereby population differentiation. Geographic distance and physical barrier enhance reproductive isolation by limiting the migration and increase genetic differentiation between population. Molecular markers also find application in aquaculture to assess loss of genetic variation in hatcheries 2. Comparison of variation between hatchery stock and wild counterparts. Genetic diversity was also investigated using microsatellite between farm and wild population of Atlantic salmon. Farm salmon show less genetic variability than natural source population in terms of allelic diversity. This information is useful to obtain in monitoring farm stock against inbreeding loss and to plan genetic upgradation program. Lastly, we have application in for assist rehabilitation program. Habitat alteration and overharvesting have contributed to the decline and disappearance of numerous natural populations. In addition, reinforcement program of wild population based on release of hatchery-reared fish of non-native origin compromise the conservation of remaining native trout resources. Effect of the program to release in a natural population has been studied in many fish through molecular marker. So, as we know from my slide, there are so many important for the research on molecular marker that can help improve computational power and get a large data available to help researchers to maintain and find new species and taxa for all animals in the world, including fish. That's all from me. Thank you for listening.